All right. Hello, everyone. Just going to wait another minute or two for people to funnel in, and then we'll kick it off. All right. Well, I'll just start with a little bit of housekeeping. First of all, everyone, welcome. Thanks so much for taking some time to be with us today. My name is Charlie Doobie, and I'm the head of regenerative partnerships at Agrology. This webinar is part of kind of an ongoing series of webinars that we have, looking at bringing in really high quality, education and expertise from world-class experts in regenerative agriculture, in carbon sequestration and carbon markets, in soil health, soil science. And today, obviously, we're going to be exploring the topic of nutrient density with Dan. A few other quick housekeeping pieces. We are recording this, so no need to be scribbling furious notes, and we will send out the recording to everyone who registered. And that'll also be on our website. We also have all the webinars that we've done on our website, and it's agrology.ag. And then we have a webinars tab. We have a lot of other really interesting ones with great experts. In terms of question and answer period, we encourage everyone to put questions they have into the Q&A, and also please chat amongst yourselves. The chatting amongst yourselves we'll do in the chat section, and then the Q&A, which you can see on the right there, that's for questions only, and that's where we'll pull the questions at the end of the segment. We're going to start with some intros, and then Dan's going to do a quick presentation. Then I'm going to ask Dan some questions, and then we're going to open it up for everyone else to ask questions. Yeah, I think the last piece that I just wanted to shout out is I personally have been attending the nutrient density, or the Bionutrient Food Association has been doing their 11th soil nutrition conference, and it's on the state of nutrient density. And there's been some amazing speakers. We were just talking with Dan about it right before we started this webinar. And so I've been really, really impressed. And given who's here at this webinar, I'd really recommend everyone check it out. The content is just extremely high quality, and the insights just the insights from the speakers, there's been quite a few mind-blowing moments for me. So I encourage everyone to check that out. With that, Dan, I'd love if you could give us a quick intro for yourself, maybe describe your journey, and I know you've been studying, nutrient density for a really long time, and you're definitely quite the expert. So how did you arrive in the place that you currently are?
Well, thank you very much, Charlie, for the opportunity to speak. Actually, this first slide here is answers your question quite viscerally. This is actually it's the front page of our website, bionutrient.org, but it's also a picture of the farm that I grew up on. So I grew up on a small holding, mixed organic farm, animals, vegetables, fruit trees, etc. My parents were actively involved in the organic farming movement from the 80s on, and so I have a long history in that community. And when I basically found no better way to live a life, or had built no other income streams, I was forced to ask some big questions about organic and farming, and that led me on this path, which is now the BFA and nutrient density and the work we've been doing. So I won't belabor the point too long. The timeline we've got strategy here for is 20 minutes of presentation, 20 minutes of questions from you, and then 20 from the audience. So let's just let me run through my presentation now, and then we can engage the conversation, which is always the most fun for me. So next slide. Yeah. We started the BFA in 2010 as a nonprofit educational organization with a mission to increase quality in the food supply. And by quality, we're talking about flavor, aroma, shelf life, pest and disease resistance, farm viability, soil health, human health. We think of that as a point where a broad social movement can coalesce that's concerned about a lot of things around systemic solutions. And we helped, coin the term nutrient density as it refers to this before 2010, probably in 2007, 2008. Next. Yeah, so the first few years of the organization were a lot of education. Basically, I downloaded a lot of information from the Acres USA community, integrated perspectives from not just organic but biological, permaculture, agroecology, conventional ag, soil food web, all kinds of indigenous cultural traditions, slow food, Weston Price. We did workshops and courses and conferences. You see up here a number of speakers that we've had at our annual conference, the one you just referred to. These are historical presenters. That was the first few years of the work, was really just sharing these understandings and say, look, oh my God, if we work more well with nature, it looks like a lot of systemic things could be improved. And as that began to occur further and further around, um, spreading basically through grassroots, word of mouth from here in the Northeast, around the country and other countries and other continents. We started looking at the systemic ways to potentially take this understanding and bring it to a larger scale. And that's where the research side began. So this next slide there. So we started in really 2016 conceptually, but 2017 actually, with a strategy of answering three questions. The first question was, does nutrient variation exist in food, and is it significant? Our hypothesis was that it did, and it was significant. And second question was, does that connect to soil health or to specific soil type or certification or variety or fertility program management practice? And then the third question was, can you build a handheld instrument which could test that variation if it was significant? And so, yeah, here this graphic is some of our preliminary results, looking at just a few elements and compounds and showing the scale of variation we found across the supply chain, which is 4x, 5x, 6x, etc. It's not small. As X means times, like this one is as much as those six is 6x, not 6%. So yeah, we started a project. We built a handheld meter. We started building labs. We asked people to send in samples of crop, and then after that, the soil that crop grew in, and answer some management data questions. So next slide. This is an example of the data we collected in 2020, which is one of those many years. You can see on the right the number of samples and the number of crops that were assessed in that 1 year. You can see every green icon is a farm that was sending in crops in triplicate with soils in triplicate at multiple depths, and as well answering a number of management questions. So we were really trying to figure out how dramatic is the variation, what are the environmental conditions that are causal and correlate. And you can see on the left the nutrients we looked at in the crop, and on the bottom and on the middle the aspects of the soil we were looking at. As well, we asked about 50 different management questions. What was the variety? When did you plant? What was your fertility program? Did you cover crop? Did you till? What was the mix of your cover crop? Et cetera, et cetera, et cetera. Next slide. Some of the core results we found, and these are just these next few slides are just about carrots, but understand we have data like this. These graphs look remarkably similar across about 20 other crops, roots, leaves, fruits, and grains. Sort of on the bottom left there, you can see 8.41. On the bottom in the middle, on the sort of on that same graphic, 33.19. That's the range in sulfur we found in carrots. So 8.41 milligrams per 100 grams was the lowest we found, and 33.19 was the highest. If you do the math, that's basically the highest one had 4 times as much as the lowest one. On the vertical axis, you can see number of samples, 0, 20, 40, and then 60 on top. So effectively what we're finding is a bell curve representation, which is a pretty good sign. But the peak of that curve is over there, and the bottom of the orange and the sort of the edge of the right, red. On the bottom, it says fifths of the range. So red is the first fifth, orange is the second fifth, yellow is the third fifth. That'll be 0 to 20 out of 100, 20 to 40 out of 100, 40 to 60, 60 to 80, and 80 to 100 out of 100. Those are the fifths of the range. And so we see that the vast majority of the sample set is showing up in basically the peak is at the 20th to the 25th percentile. Almost all the carrots we found no, we're gonna stay on that slide for a second longer. Um, we're relatively low in relationship to what they could be in this one element, critical element for human health, which is how we determined which nutrients we're gonna look at. On the right, phosphorus 11.7 to 93.69. That's an 8x variation. The ones on the right had 8 times as much phosphorus as the ones on the left. And again, the peak of the curve is in that 25th to 30th percentile. Keep going. Antioxidants, those are very, very health-giving compounds. 4.92 on the bottom left there and 195.05. That's a 40x variation, as in the ones on the right had 40 times as many antioxidants as the one on the left. And the peak of the curve is in that, 15th to 25th percentile. Not high in relation to what's possible. Almost everything we found in the supply chain is relatively low in relation to what it could have. Polyphenols on the right, 2.34 to 49.75. That's a 20x variation. And the peak of the curve is there in the 10th percentile, 5th to 10th percentile. So, this is again, more than 20 crops, many years, multiple continents, hundreds of farms, all kinds of soil types, varieties, management practices, certification labels. This is the nature of the variation of the supply chain. There's a significant variation in nutrient levels, and most of what's available is relatively low in relation to what could be. So, next slide. On the left here, we have by variety and BQI score in carrot. That's an average of 6 nutrients and 6 elements in two compounds. So some people say it's variety that matters. And we see here Napoli on the top, and then Bolero, and then Nantes, and then Mokum, etc., Romance. You see a number of different varieties and no direct correlation between that variety and the average, score of nutrients being high or low. We see a nice dispersion of variation there. So it doesn't look like variety is what matters. The second slide on the right, by climate region, those are USDA climate regions, Northeast, East, North Central, Southeast, Central, etc. Again, variation across the board. You can do a bad job in any climate zone, soil type, or a good job. It's not the soil or the climate that seems to be what's correlating. Next slide. This is by sample source. So a lot of people in the movement will talk about where you get your food and how that correlates to nutritional value. This slide has a bunch of uncomfortable facts in it for some people. First is from farm, so get it from the local farm. Somebody's doing a good job. I'm growing it. I'm a farmer. Bunch of reds, not too many dark greens. Store is next. Actually, looks like the spot where it's fattest on store is to the right of where it's fattest on farm. Not comfortable. Garden is the next one, and then farmer's market is the last one there. So we're not seeing that because you have a garden or because you're a farmer or because you buy at a farmer's market, necessarily the ambient level of nutrients in the crops you're purchasing is higher. What we found was most things are relatively low in relationship to what they could be. On the right, certified organic on top. A lot of reds, even more oranges, just a couple dark greens. People who use cover crops there as the next one. Looks a little bit better on average than certified organic, actually. No-till as well, better than certified organic, but still the center of the curve is between about 40th percentile, the fattest spot. People who use tillage, not really that much worse. Organic, not certified, same range. Reference and none, those are actually grocery store samples that are not organic, and where they're fattest is a little bit to the right of these other things. So some farmers on scale actually not doing that bad of a job, looks like. We're just farmers. Regenerative is the next one there. That fattest spot there is definitely below reference and none. That's self-identified regenerative, biodynamic, local, etc. So really across the board, we're not seeing connections between any individual practice, certification label, and quality. The graph I really need to get into this presentation is the one where we showed that the only thing that correlated was the level of life in the soil. It seemed that only where farmers were actually doing a good job managing a high level of life in the soil, were we seeing increased levels of nutrients? That really is the biggest takeaway here, and I think probably where our conversation will go after my presentation. Next slide. So in 2021, we released this instrument, the bionutrient meter, handheld, consumer priced flash of light meter that you can use in real time to assess relative nutrient levels in crops. We proved, at this point, basically our three questions in the affirmative. Nutrient variation is significant. It does connect to soil health, not other sort of individual factors. Overall soil health, it seems like, if soil life can be a stand-in for soil health, and it's possible to build a handheld consumer-priced flash of light meter that can assess nutrient variation in crops. So that's the sort of the preliminary work completed, and then the last couple slides are on our current work with beef. Just go to the next slide. Yeah, so basically after we determined that variation is significant and meters can be built, and it connects to soil health, I thought was, great, well, let's use this then as a strategy to organize a broader social movement around this question of nutrient density. And if we can support people in buying food that's better for themselves and their families, and it's also better for the environment, then we can use the economic power of our daily purchases to potentially have very meaningful and rapid positive impacts on the planet, culture, etc. So to do that, we need to actually have a meter that tests overall quality, not just polyphenols or antioxidants. And so to do that, you have to define what overall quality is. And so we've been working for the last 3 years to define nutrient density. We chose beef as that crop to work on. You can see basically the thesis here, which was that the greater the diversity of the foodstuffs that the animal has access to, the better the quality of its meat will be. So next slide. This is sorry, it's not great. Can't quite see the details of some of the text here, but we're looking at hundreds of different nutrients in every piece of meat. We're looking at hundreds of different species in every manure sample for the microbiome assessment, deeply into forage, deeply into soil, all kinds of management questions being asked, and then as well doing some human health trials. Really excited about this project, which is hopefully will be completed by the end of this year. Next slide. You can see the geographic diversity of samples, primarily North America, but a broad diversity of climate regions. We have Europe, we have South America, get more in Australia than you can see there. Keep going. Preliminary data, if we can use the omega-6, omega-3 ratio as a stand-in for quality, which is sure what it looks like it's going to end up being, that'll be a significant component of it. Dramatic variations in how the animals are fed in their last phase of life and nutrient levels in their meat. So this omega-6, omega-3 ratio is understood to be really powerfully connected to human health, chronic disease, inflammation, all kinds of really important things. And I think American Heart Association says more than 4 to 1 is bad. Generally below 2.5 to 1 is good. You can see the average in the grass-fed is 1.7 to 1. Average in the grain finished is 7.8 to 1 there. You can do a better job or worse job in each, of course, of these areas. It's not a black and white. Some of those outliers that are on top on the left on the grass-fed section actually were claimed grass-fed, but we didn't have any management data, so we think we can probably tell that those claims are not correct, or technically maybe those cows did eat grass at some point in their life, but not in their end of life. Last slide. And again, preliminary data will only have this available in a sort of published format next year, but it does again sure look like the level of diversity in the ecosystem that the animals have access to, the better those underlying nutrient levels in this case, the meat that we harvest from them. So that should be a quick overview of our work. I wanted to respect our time boundaries, and I think I've kept it to the first 20 minutes. So yeah, I see a bunch of questions in the Q&A. Let's see where people want to go.
Yeah, amazing. Thanks so much, Dan. And thanks. I know that that could have stretched out for an hour and a half. So thanks for trying to keep it packing a lot into a little time. I'm going to start out with a few questions that kind of just occurred to me while you were presenting. One is, so we saw obviously there's like a much larger spread in the compounds like polyphenols and antioxidants, which I believe are secondary plant metabolites,
What they're
versus
called.
the actual
Yep.
nutrients like sulfur and phosphorus. Why do you think that there's more of a spread when it comes to those compounds?
Good question. I think basically the elements are considered to be necessary for life. The secondary metabolites are not necessary. They're called secondary because they're not primary. Primary means you must have them to not be dead. Um, so
Yeah.
one could argue the greater your health, you can really start pushing the envelope on very high levels of these things, but you can still be alive and have very, very low levels. So yeah, the elements we learned about in chemistry
So,
in high
so it's
school.
almost like that's,
Yeah, sorry.
go ahead.
Well, just the
I
elements
was just going to say
are foundational. Yeah.
it makes sense. It makes sense to me that the secondary plant metabolites would be sort of a more sensitive measure of soil health, right? Because they only can occur, as John Kemp often talks about, kind of at the top of the pyramid, right? When you have all of the other nutritional needs are met, then the plant can start to create those. So I guess it would make sense to me that you could see potentially more of a spread there. And then another question we got in the Q&A, and I'm also curious, why do you think that there was different spreads in different crops? So for example, we have a lot of grape growers in the audience. Why do you think grapes were kind of on the higher end with a 15x or more spread compared to other crops where it was only maybe 1 or 2x?
That's a good question. I think, the scale of our database is so large and our scale of our funding is so low that a lot of these questions that could be answered haven't been answered yet because we simply haven't had the bandwidth to delve into them. Um,
Yeah.
yeah, I mean, the general point was that, what we were told by people who, presumed to know better when we began this process was that it was a 2% or 5% or maybe 10% variation from top to bottom in any of these crops. And we found something much more dramatic than that. Yeah.
Yeah, absolutely. I was really surprised when I ran into this data set and kind of the results, actually it was just a few months ago. I was really blown away and also really excited to see that soil health was really the only thing in soil life that was correlated with increased nutrient density. Kind of building off of that, why did you guys choose respiration as the way to measure microbial activity or kind of soil biological activity? And then what method did you use for measuring soil respiration?
We chose it because we didn't have much money and it was a good stand-in for overall biological system function, which is a very complex thing, which we don't really understand. It was some version of the Solvita test. It wasn't the Solvita test, but basically we take dry soil in this case, a certain amount, add a certain amount of water, put it in a box at a certain temperature, and measure the amount of carbon dioxide emitted in 12 and 24 hours. So very simplistic, very inexpensive, able to sample again from hundreds of farms from multiple continents at a very low cost point. So, but
In
we thought a really good sort of stand-in for overall system function.
Yeah, I mean, I think that there's a large body of research that would agree with that, right? Obviously, we know soil respiration is sort of an oversimplification of the
Yeah.
amazing web of life that is in soil. But I always kind of describe it as it's like listening to the amount of volume coming out of a party. It's like a pretty good indicator of how good the party's going to be.
Nice.
Although you might get in there, you might be surprised who's there.
Yeah.
Speaking of that, are you going to be doing kind of any metagenomic analysis in future research and kind of diving in and understanding those functional communities? Or do you think you'll continue with just monitoring the volume of the party from the outside?
With the beef project, we're looking at, like I said, hundreds of different species of multiple kingdoms and levels, etc. I think this is a field which is, you know, how much do we understand? 2%, 5%? I mean, we're beginning to find some basic patterns, but I'm happy to take the lead of those who know more and, you know, try to run 3 or 4 different modes of assessment against each other and see what works most well. Um, what it sure looks like is this is an important place to focus our attention in further research. There's something really important going on here, which is, you know, how the microbiome is functioning. And, you know, I think we probably hypothesized that. I think perhaps more and more of the conventional agronomic community is coming around to that, but historically that was not part of the generally assumed state of things. So at least we've perhaps helped to, you know, support that
Yeah.
understanding and support that intention to look more deeply into it.
Yeah, and again, I think that's probably also a function of just where we're at, sort of in terms of technological capacity. I always really love John Kempf's line about how we are only really looking at chemistry because we can only easily measure chemistry, and now we do have the tools to more easily and more thoroughly measure biology, microbial function, all these different functional communities, and so now we can actually use these tools to better understand
Yeah.
something. As you said, it's like deep space. Maybe we understand 2%, maybe 5%, but it's pretty
Yeah.
exciting to think about the meta data sets that your organization has pulled together, that all of these different groups are kind of starting to gather, and especially applying machine learning and AI.
Yeah.
It's going to be a really interesting next few years, and I think and looking back in 5 years, we're going to understand a whole lot more about the soil microbiome.
And the real question is how much of that is done openly and collaboratively, and how much of that is done proprietarily. And can we get a critical mass of, what do they call it? What's Christine Jones calls it? A quorum sense, where
Yeah.
enough of us are coming from different parts of the ecosystem and starting to work together that we're building this collective where we really can transform understanding. Because I think that the danger is that people are going to try to, black box it. But yes, either way, we're going to know a hell of a lot more in 5 years. Um, and it looks like it'll be very, very empowering. Yeah.
In a classic universe is a giant fractal moment, that quorum sensing to me really reminds me of the soil microbiome, right? That's
Yeah,
exactly kind
exactly.
and so
Are we going to behave that way or not?
yeah,
That's the
exactly. Are
question.
we going to
It's
learn?
up to
We're
us
going
to
to learn
choose
that actually
to be
not just study
symbiotic
it,
or
but
not.
act like the soil microbiome.
Yeah.
Then I
Well,
think that'll be
it
really,
acts pathogenically or
yeah.
symbiotically depending on environmental conditions. So we will act like the soil microbiome. It's just a question of in what fashion.
All right. Now we're getting to like the meta of the meta layer. Super fun.
I don't know,
So
but
to
yeah,
kind of dive back into some of the research and some of the findings that you had. So obviously your research did not find correlations between, organic and regenerative certifications and increased nutrient density.
yeah,
I think we all know that the premise of these certifications is absolutely valid and worthwhile, right? We know that to some degree we need to standardize for supply chain adoption and sort of simplify for consumers to be able to easily understand on the grocery store shelf how we qualify someone as regenerative or organic or whatever. So if we're looking at your findings and we all agree that regenerative agriculture needs to be based on outcomes like increased soil life and improved, nutrient quality, how do we balance that with the need to standardize this stuff so that it can be adopted into the supply chain and that consumers can easily understand it?
well, I think you build standards that are not binaries, but that are continuums. And so instead of having a regenerative, not regenerative, or organic, not organic, you've got a 20, 40, 60, and 80 out of 100. And you can
Yeah.
establish a standard that says this is 1, this is 100, and this wheat is 20, this wheat is 40. But I think the only real way to do it is to get out of that. We can call it a reductionist, colonized, mechanistic, binary, you know, um, non-life frame. So I think it's been a significant part of what I've been trying to accomplish after having watched my parents write some of the first organic standards in the country in the 80s and having watched that whole system get, I would say, perverted to a large degree. Still is an immense amount of value in the organic label, but in relation to what it could be or could have been, I don't think it has come close to arriving. Um, and I think we need to unposit this practice-based bureaucratic certification framework and engage a continuum-based empirical framework. And if it's
Yeah.
true that soil health connects to nutrient levels, then why not use the relative nutrient level as the thing to determine where something sits in the continuum? Um,
Yeah.
let
Yeah.
markets evolve. I'll take some 60s and some 40s, but I'm better than my competition, which took 40s and 20s. and
Mhm.
it's constantly evolving better. As we can see from the data, there's not a lot of 60s available. And so people that are operating on scale, you know, um, you're gonna have a hard time sourcing the amount of quantity that's at a good level, if you want to make nutritional claims. So yeah, it's going to be a dance, I think. But I think the more data we have, the more open it is, the more well it'll the process will yeah, occur.
Yeah. That answer definitely resonates with me, and I think is very much in line with kind of what we're also pushing for at agrology. And I think we're seeing that emerging consensus, right? That regenerative agriculture needs to be outcomes based, and it needs to be not just a line in the sand where it's it's practice based, and you're either in the cool kid club or you're out. But really taking that Cindy Daly and Tim Lasal always talk about the big tent approach, and we're big fans of that. And I think that, like you said, let the data speak for itself. We now have the ability to measure soil respiration continuously, pretty cheaply on farms. We now with your new, the nutrient meter, we have the ability to measure nutrient density. And I think that oftentimes there's sort of we think too low of consumers' ability to discern. We think, oh well, they'll only be able to understand, like it's either organic or it's not. But I think that I think that there is a group of consumers that are a little bit more discerning and would be very excited about seeing a spectrum and supporting, the things that are higher on that spectrum. Or, maybe they always want their carrots to be really high end, but they don't really care about, their broccoli. They'd be happy with a 20. Um, so next question is, as we all know, or this was always my line when I was running farms, is irrigation isn't irrigation, right? How we apply a practice matters perhaps as much as the practice itself. And so we know that there's these principles, practices of regenerative agriculture that can really boost soil biologic function and soil health. But that's not always the case. Just because someone is employing cover crops doesn't mean that they're choosing the right seed blend or planting it at the right moment, or getting a good stand. And oftentimes too, people are in these practices, or they're just assumed to work. How can we build a better data set and a better intelligence for growers to understand, not only the practices they need to deploy, but how to deploy them and sort of how to like dial in their regenerative program? Do you think
Yeah.
like a KPI like nutrient density could be something that people really center around, or respiration, or what sort of KPIs would you use?
Well, I think the result, hopefully, is nutrient density if we can define it well and have instruments around that definition that can be used in the market. But what's most important for the producer is the real time modulations and the practices in season and the planning in the beginning of the season. So I think you again mentioned our conference. We just had a presenter on yesterday, David Knaus, who's out in Oregon, who's been doing brilliant work with apical ag in really digesting like what are the 8 different forms of soil tests and how can we use 5 of them to build a completely systemic perspective of what is going on and what needs to be going on and how you can use 1 pound of micronized humates and 2 pounds of micronized rock dust to transform a underlying, sodium excess
And
or aluminum excess, right? I mean, we actually do now have the capacity from integrated people who are pushing the envelope from an agronomic perspective. I mean, you obviously John Kemp is famous in this world and rightly so, but there's a lot of people out there who really have the capacity to support systemic transformation in short order based on empirical data. Um, and I would say if I could offer a critique perhaps of the regenerative community, which I think is certainly due to the organic community also needs to hear this, where I come from, is there is a blend of really deep science, proper, hard, published, lab science that has been built over the past number of decades, which can be completely empowering to organic producers and completely empowering to regenerative producers. I think the sort of like the 6 regenerative principles, living roots and living cover and animal integration and don't disturb the soil. Those are all sort of grand overarching statements, but in most cases, there's a level of nuance which we have access to now, um, which is completely empowering. And I say that came from the Acres community. That's where I accessed it, and a lot of the people that I'm working with that I think are doing the most well um, came up in Acres. Now is different than Acres was 30 years ago, obviously, but yeah, there's a lot of opportunity there to use empiricism and good lab work at the beginning of the season to really just get much better results. So certainly at the end of the day, I think
Yeah.
my vision is that nutrient density is a market metric. I do think that respiration is, completely correlated. And as far as like look at one thing that's sort of where do things stand? It's a great way of looking at it. But to really cause a dynamic where that occurs well requires a level of insight that is very available. So that's a long
Yeah.
answer, but
I absolutely, I love that. No, that was a great answer. I love that. Right? We need the expertise. We need the ability to kind of take, both point in time, but also continuous data, continuous respiration, but also I'm super excited to see sort of, I believe it's a similar technology to the nutrient meter that you have for final crops that John Kempf is releasing for kind of real-time SAP analysis.
mhm.
It's like, I always believe that anytime we measure anything, there's just inherent human flaw in it, right? It's not the whole picture. But if we can measure things through a variety of different approaches, metagenomics, real-time SAP analysis, nutrient density measures, I think we can start to paint a little bit more of a picture. But I love your point that that's actually not enough, right? We also need that wisdom, really. I mean, it's expertise and wisdom, and that agronomic expertise is available in the world.
Yep.
And so, yeah, it's
In a
a
way that it wasn't
really
5 years
exciting
ago,
moment.
or even 2 or 3 years ago. But it really is coming online now in a way it hasn't happened before, and it's extremely exciting. Yeah, yeah.
Absolutely. Um, so one other question I had, and I've got a few more, and then we're gonna move it over to the open Q&A. Can you tell us a little bit more about your handheld nutrient density meter? I know you talked about it briefly. I heard, and I think it was a podcast you had with John, and you described how spectroscopy worked, and it was like with starlight, basically, was the metaphor, or looking at a star. And so I really liked that explanation because I hadn't been able to understand how that technology works
Yeah,
before. So yeah, can
yeah,
you
yeah.
just give us a little bit of the science behind how does the handheld nutrient density meter work?
And you've said nutrient density a few times and referring to our meter as being able to test it. And I just want to make a small technical point, which is um, we don't know what nutrient density is yet. So our meter currently does not test nutrient density. That's the work we've been trying to do with beef, and we hope to do going forward as the next level of our work is to define nutrient density so we can build meters that can test for it. So the meter we built so far only tests for certain individual compounds. I try to say this every single time, but people say what you said, which is Dan's meter tests nutrient density. No, it doesn't. We don't even know what nutrient density is yet, so
Yeah,
we can't measure it.
I'm just gonna
We've defined it.
oversimplify and just put it in a
Yeah,
nice
yeah,
little
yeah. Well, that's the
package.
problem with like bricks measure sugar. No, bricks does not measure sugar. Um, yeah. So um, basically every element in chemistry that we might have learned, in, I don't know, 10th grade has protons and neutrons and electrons. Um, in physics is a vibration, is a vibration of light. And so the way that we in science and astrophysics know what the sun is made up of or what Alpha Centauri is made up of, or if you ask anybody studies the stars, they'll tell you these stars are made up of that, and we found methane on this planet over here. And how do they know that? Because we've never shipped a scientific probe off to a planet, off to a star, and sampled it and brought it back. It's because every element in chemistry vibrates in physics, and so copper has a certain vibration, and zinc has a different vibration, and so that vibration is light, it's color, and so basically, if you're really good, you can take a picture of the light coming off of a star, and you can see what it's made up of. And so that's all we're doing is we're taking a picture of the light coming off of the carrot or the cucumber or the steak or the milk, and we can actually see what it's made up of by the fact that everything in chemistry, a copper or zinc or protein or polyphenol or fatty acid, is a vibration in physics. And so that's the technology. It's called spectroscopy. It's been around for decades. And it's the means by which we propose to establish this new market standard of people being able to assess nutrient levels in food at point of purchase, non-invasively before you walk to the grocery store shelf, you say beep, 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 and 3 different options, and one's 20, one's 40, one's 60 out of the 100, and you can choose accordingly. That's how it works. It's not the QR code, it's not the marketing, it's not the labeling, it's the actual testing through
Mm.
spectroscopy of the nutrients in the food itself. Yeah, it's pretty much like Star Trek. It's pretty cool. But so are smartphones,
It's amazing.
so
It's true. So you just mentioned polyphenols, and I know that we have a lot of grape growers here, and so yeah, really curious if we could talk a little bit about um, phenolic compounds. So essentially, we're studying nutrient density, or rather concentrations of minerals and certain compounds in both crops and now in beef and or in milk. How do these relate to the phenolic compounds or the secondary plant metabolites in wine? And can you maybe draw some? I know that from my research and talking to a lot of winemakers, those are the things that lead when you have high density of those phenolic compounds, you have higher quality wines. So how can you kind of translate all that you've been researching in food crops into vineyards and in wine production?
I can call grapes food and tell you what I understand about grapes. I'm not sure I know much about
Perfect.
wine at all, but it sure looks like those things we call plant secondary metabolites are very, very, very large compounds. They're built with tens of thousands of different elements. They're really big things. And a um, the way nature has evolved the ability to build those is through soil life. It's through microbes, put in the first 20, 50, 500,000, 10,000 compound chunk together, and then shipping that up through the plant where it can use it wherever it wants. And so it looks like basically the greater the level of life in the soil, the greater the capacity to have those secondary metabolites in the plant is. Um, Now, to what degree does stress in what form affect that level, and how does it relate to yield, and how does it relate to other nutrients in the plant? I'm not sophisticated enough in my knowledge of the science to be able to speak to that, but I know that foundationally you cannot get high levels of those compounds without a well-functioning soil microbiome. It's just a simple fact of take an ion of this and an ion of that and put them together, you got two, and then you got to do that again, you get four, and then you got to do that again, you got six, and to get to 10,000 elements in a compound is a lot of work. It takes a lot of biochemical work. The plant doesn't have that much energy. That system only really functions in the soil with the microbiome. So yeah, best I've got.
Amazing. And then I know, from my own experience and also from working with a lot of growers, we also know that when you have high levels of these secondary plant metabolites, you have plant disease resistance,
Yeah,
stress resilience, fungal pressures literally cannot eat those fruits. Can you
exactly.
describe why that is? Because sometimes, like when I first started learning about this, I thought, oh, well, if it was a more nutritious crop for humans, then it's probably more nutritious for the pest. But it's actually the opposite effect.
Right. Yeah. So when I'm teaching my course, I use the example of a bale of hay, and I say, if there's 20 of you sitting here in a room, or 50 or 100, whatever it is, and when you walked in this morning, instead of it all being chairs, there was a couple bales of hay. You might have considered sitting on one of those bales of hay, but you probably would not have considered eating that bale of hay. And people say, yeah, yeah, that's true. And I say, and theoretically, if someone had walked some cows in instead of humans, those cows would not have considered sitting on that hay, but they might have considered eating it. People say, yeah, that's true also. Um, and I say, why is hay food for a cow and not food for a human? And they say, well, because cows have four stomachs and they can digest complex cellulose and we can't. I'm like, yeah, that's right. Okay. So if you grant that point, you understand that different organisms have different levels of digestive system function. Um, and yeah, all larvae don't have livers, right? They don't have the enzymes in their gut to digest protein. So when your plant through good soil life symbiotic relationships is able to complex complete protein out of amino acids, it becomes indigestible to larvae. Um, and so we know about the soil-borne pathogens have a hard time digesting carbohydrates, and with the um, the blights and the mildews, they can't digest through strong levels of lipids, the fats and oils. And then the beetles, when you start to get the secondary metabolites, they can't digest it. So what I say is, if you're trying to grow animal food, and of course insects are animals, but say warm-blooded animal food, um, you are only, if nature is sending in other organisms to eat your crops, what nature's telling you is those crops are fit for those organisms. If it's fungi that are eating your plants, then nature's saying this is fit for fungus to eat. If it's larvae eating your plants, nature's saying it's fit for larvae to eat. Only when you have animals as pests, should you understand that nature's telling you you are properly going warm-blooded animal food. So, yeah, there's a whole bunch of science behind that, but that's hopefully a metaphor that sort of can convey the concept.
Yeah, no, you conveyed it beautifully. Okay, I'm gonna start jumping into some of the questions in the Q&A now. One we have from my colleague Jay, actually, and I think it's a good one, so I'm gonna start with that, although I'm a little biased. He says, while we know we need to eat more whole foods, the reality of the American diet is that it will always probably be partially processed. So do these nutrient results or higher nutrient density carry through to food products that typically come in a box, a bag, or a bottle? And if that hasn't been studied yet, like what's your guess or what do you think?
Depends on how you do it. You can take wheat of high quality and you can take the bran off of it and turn it into white flour and put some chemistry in it, which will cause it to expand fast and cook, and you can cook it fast and you can call it bread. Or you can take wheat and you can sprout it and grind it and let it sourdough ferment for a couple of days and then cook it. And you can have very, very, very bioavailable, positive nutritional thing here and not so much over here. So post-harvest processing is massively important, and it can improve or degrade inherent nutrient level. Yeah, it's a big piece of the puzzle. And again, back to the conference we're talking about, with
Yeah.
Westgate from Non-GMO Project. They're, the people who've made that butterfly seal are working systemically on a process by which we can actually have a label that correlates with it was not only grown well, but also processed well. Wouldn't that be nice? Wouldn't that be nice if you could say, like, this was lacto fermented and this was full of nitrates and preservatives? They're both salami, you know, like all these traditional foods that we have now in our sort of corporate versions
Yeah.
came from somewhere that might not have been that bad. A donut made with whole grain and fried in lard that was, from a healthy pig. Like, I don't think it's that bad actually. A donut proper. Um, yeah. So it's really about how you do it. It's not the cow, it's the how.
Yeah, absolutely. I love the places we're going on this webinar. Um, so kind of following on that question, how do we get food industry, CPGs, retailers, etc., to care about nutrient density? We know they valued organic. We're starting to see they're starting to put value on regenerative certifications. Where are you seeing traction for nutrient density, and what needs to happen for there to be more traction across supply chains?
Feels like the buzz is building pretty well right now. I'm getting called globally to engage people of all kinds of different scales. It's one of those things where it takes a little bit of time for the word to get going and let the data start to get there and people start to check it out and they think about it and they talk to somebody else, and it feels like we're on a strong yeah, upswell, a wave. There's a big wave coming right now in the topic. For me,
Yeah.
the way you get the CPG attention is with the market, and that's why you have to have the instrument, the meter, and you ideally want to support the market with, here's how you can do a better job, and here's when the meter's coming out, and here's a expensive meter that we'll have first before the consumers have their meter, so you can check your supply chain. You get 6 months of lead time. Here's how you can work with your producers, if you actually know your producers, to ensure they're all doing a really good job, because that's all available. It's the carrot and the stick. The stick at the end of it is consumers are going to have the ability to test, and the carrot is we already know how to do a better job. which is better economics and way better storytelling. So why not jump in now? And I feel like that's the dance we're in right now. It's very exciting. It really feels like it's got a lot of momentum. Um,
Are there certain supply chains where you've seen kind of a little bit more traction, or like they're a few steps ahead?
well, I mean, we've been working with beef because that's an area where there are a lot of people doing a good job and differentiating themselves because of the good job they've been doing, and they're looking for the ability to formally say, "Look, our stuff is better." So there's many dozens and dozens of producers around the planet. I mean, thousands, I'm sure, but dozens we've been working with, many dozens. Um, yeah, I mean, I haven't looked so much into coffee and wine and things like that, where I say people pay more attention to their drugs than they do their food. So, I mean, there's a lot of discernment around coffee and flavor and wine and flavor, and it does connect. It does totally connect. But let's have, I mean, yeah, we haven't gone there so much. We've been focusing on the foodstuffs, rice and wheat and milk and apples and beef and stuff like that. Um, but I feel
Yeah,
like the whole, I feel
yeah,
like the,
no,
I feel like there's a lot of bubbling up going on in all kinds of different, um, yeah, pieces of the supply chain.
perfect answer. This is from our friend Tom Willie. He asks, how well do your findings agree with other similar efforts around the globe? And can you discuss other groups that are investigating nutrient density? And why has it taken so long for these inquiries to get traction?
There's three questions there. Give me one at a time. People around the globe who are doing it. Um,
Yeah. How well do your findings kind of agree with or correlate with other similar efforts around the globe? And so who are these other people around the globe?
the problem is I'm not aware of anybody looking at, management overlaid against soil, overlaid against variety, overlaid against microbiome, overlaid against biochemistry, overlaid against human health. That's the thing. Like to define nutrient density, our thought is you find the pattern where all those things connect. And there's people looking at microbiome, there's people looking at human health, there's people looking at management practices, there's people looking at genetics, people looking at soil types. Um, um, yeah, I mean, I would love to say that there's groups that have been doing something more significant. I mean, there's Blue Blancoeur in France that did a lot of work, 20, 30 years ago, looking at omega-6, omega-3 ratios in animal products and how animals were fed. And they did some human health trials, which showed very positive things. I don't think it was anywhere near as nuanced as what we've done, but certainly great work. There's PTFI, which is starting up, and that's mega funded by American Heart Association and USDA and Rockefeller Foundation, and they're looking at interesting stuff. There's groups around, but as far as there being a cogent movement, I mean, obviously we've been working with Chico State and what they've been doing. They've been core partners since early on. Um, yeah, I would say we're a very nascent community. If you look at any one of the pieces of the data, does what we found seem reasonable? Yes. The nutrient variation, if you actually look at the nutrient variation data, that is significant. If management connections to results, that's yes. So I would say that our data is verified by a lot of the pieces that are out there, but the way we've been trying to go about doing it, which is, build this sort of integrated framework, hasn't been done. I don't know if I got all of his
Yeah,
questions.
you'd have to be just the right amount of crazy to look at all of those different factors. And I think that you guys have that wild, what does they call it? Crazy wisdom to look at such a holistic approach, right?
The point is, it's nature, and you can't just look at the soil minerals and say you're understanding nature, right? You can't just look at the microbiome and say you're understanding nature. You got to look at all these pieces together. And that's really been our effort, is to say, where do we get our guidance from? And like, who knows what the answer is? And our thought is nature knows the answer. And we're using the scientific method to see if we can garner wisdom from nature. But we have to do that by looking at all these different components in relationship to each other and finding the patterns, letting the patterns guide us. It's original research, right? It's not like,
Oh man. Yeah.
yeah,
I love it. That was my favorite answer that you've given all day right there. And kind of dovetailing on that, right? Kicking down the door of Newtonian reductionism and looking to nature for patterns, but using science to sort of find these patterns and using those to illuminate our practices.
yeah.
I have a question here from my friend Martin. How are you defining regenerative? And if you could, how do you think you're sort of defining it currently in your research? And if you could redefine it in some way for sort of the growing church or the growing big tent
Yeah,
of regenerative, how would
yeah.
you define it?
Nice to see you there, Martin. Yes, when it came to our research, we asked people to self-identify. So you identified as organic, you identified as regenerative, you identified as no-till, you identified as certified organic. And so in this case, it was simply people who identified as regenerative. It wasn't even necessarily people who were certified. Um, and what we found was basically the general dispersal of success as we found everywhere else. What is regenerative? It is biological, it is agroecological, it is indigenous, it is symbiotic. Um, I think I mean I've really tried to stay away from using specific words or trying to define things clearly because I again saw what happened to organic when I was a kid and my parents were working for the organic movement and how that impulse got perverted. And so um I'm certainly not wanting to have a in my vision, you don't have a you are regenerative, you aren't regenerative. In my vision, it's a continuum of less to more. And that can be
Bingo.
verified through empiricism. It can be verified through all kinds of ways, but I mean, the biodynamic people always say it's a process. That's what the biodynamic community says. And I think it's as much about the internal, um, your own way of being and your own psychological, emotional status and your emotional relationships, your personal relationships, as much as it is anything else, your cultural health. I mean, I think the people in the regenerative movement that I have most respect for are doing it in a deep way in the structure of their companies, their personal lives, um, their communities, and the level of soil carbon is, one of many data points, but not actually. It's something much deeper than that.
Beautifully said, Dan. Couldn't agree more. Yeah, I think there's a lot. Like I almost want to just let that one kind of echo in the chamber for a second because I think that absolutely regenerative agriculture is it at its essence is it's you can't just define it and put it in a box. Number one, it's like putting wind in a box. As soon as you put the wind in the box, it's no longer wind. It's
Yeah,
process, it's energy. And also, I love the nod to the internal process, right? That it's all so how you run your business, how you treat your employees, and the relationships. Really, I love not to keep quoting John Kemp, but I have a hard time not doing it. I love that he points out it's all about relationships, bringing relationships back into the supply chain, bringing
yeah,
relationships back between, and that's like where for me that's the most exciting part about agrology is like how can we empower the relationship between the farmer and the soil microbiome by
yeah.
allowing better dialogue between those two, allowing kind of a more cohesive loop. Um,
Beautiful.
quick question. This one should be easy for you. When someone in the Q&A is asking, is your meter commercially available for consumers?
Nope. Because it does not test nutrient density, and because that's what people want, even though we told them it doesn't do it, we stopped making them because they kept ordering them and saying, "Hey, this doesn't do what I want it to do." Like, we told you it doesn't do what it doesn't do what we want it to do either. All it is showing it's possible. Like, it's possible to have a handheld ray gun, like straight out of Star Trek, that can tell you things. We had to prove that. And we had to make a few hundred of them and ship them around the world and have it all be scientifically verifiable before we knew our vision was possible. Like, we could actually accomplish this vision of, you know, completely transforming agriculture through empiricism and transparency and nutrient density. So yeah, if you are interested in our work, bionutrient.org is our website. Please feel free, if you I think you spend more than a few seconds there, there's a little pop-up that says, give us your email and you can get on our newsletter list. Yeah, there's a lot coming down the pike as far as meters that are coming. Right now, nothing is available that we're selling per se beyond the refractometer, which is 195 years old. The specs are there to build the one if you want to make 1,000 copies of the one we made, the specs, you can download them and make them yourself. It's open source. Everything we've done is open source in the commons. But No is the simple answer.
Well, yeah. Also, I want to give a serious hat tip to open sourcing everything. Again, not allowing sort of the need to turn everything into IP or to profit. I think that the open source approach is a really regenerative approach. So yeah, kudos for
Haters
that.
pattern.
Yeah. One kind of question that was occurring to me here is, obviously we know we haven't really talked much about carbon sequestration or climate change. And obviously we know that regenerative agriculture does a whole lot more when well applied, right? These regenerative principles, when well applied, it does a lot more than sequester carbon. But kind of with that said, how can consumers or CPGs or retailers even investing in crops with increased nutrient density, or how can we use nutrient density as a lever sort of to act on climate change? Can you tie that whole sort of thing together for us?
Yeah, well, I think I mean climate change is a whole other conversation I'm not sure we want to get into. But as far as like the modes by which I understand that humans can have most climate balancing effect, um, it is working with the land in a way to bring it to as much greenness and diversity and perenniality, et cetera, as possible. Um, that's going to affect the water cycle, which has a very, very positive effect on time on temperature. Multi-Yenna's work, for those who are not aware, is pretty clear on this. Um, yeah, it sure looks like the greater the biodiversity, the greater the microbial diversity, the greater the nutrient levels. And so if it's true that, you know, that diversity correlates with climate resilience and that also correlates with nutritional levels, then I think one of the most powerfully positive things that anyone can do for the climate and the environment broadly is to feed themselves and their children or their families or whoever else they're feeding the highest quality food as possible. I mean, I don't see any better thing you can do. I don't think buying a Prius or a Tesla is anywhere near as powerful, in all honesty.
Yeah, I I, I like that answer. I think probably a lot of the farmers, here in the audience do as well. I'm gonna just pop over to the Q&A again, see if we've got any more questions. Let's see. Well, we've got another one from Tom. And it's a good one. So he's asking, is defining and measuring soil health just as complex as defining and measuring nutrient densities in products grown in the soil?
I would say that there's no um, agreed upon definition of soil health that I'm aware of. Yeah, I would say we're in process. And hopefully it's an iterative process where we don't come up with an answer in 3 years and say this is the answer, but we have like maybe even a coalition of congresses of people that are constantly iterating and evolving that definition, which hopefully is how it will work with nutrient density as well. We may come down to some pretty key points, but let's keep it alive. Let's not have it be a fixed thing. But to the point specifically, I don't think there is a agreed upon definition. There's not that I'm aware of a definition of soil health. Yeah, it's totally in live process of evolving.
Yeah. Um, I, I guess just kind of looking through the questions here. I want to make sure that I get to all of them. Let's see. Well, we just got one in the chat here. Not really. Let's. It says, "Have you guys put any thought into rationalizing health of the soil and the quality of food and maximizing business process, like a formula or something?" Not exactly sure what that formula would look like, but I guess just sort of riffing on that too. Can you talk about how bring us like 5 years or 10 years down the line, if this movement continues to grow and things go well and we continue to think regeneratively,
Yeah.
how can you see markets shifting where growers are growing for actually nutrient quality and density as opposed to just yield? And how are you seeing profitability sort of tied into increasing nutrient quality?
I mean, that's a whole half-hour conversation at least. what we're looking at right now after completing the beef project is animal agriculture, and looks like we have some basic, biomarkers that seem to correlate. And, how about if we could shift the way that all animals that are raised for human consumption are fed in the next 5 years? Let's imagine that pigs and chickens and cows are eating diets that are appropriate for them, and maybe even living lifestyles that are appropriate for them. We wouldn't actually have the need for all the corn and the soy to be produced in the first place. The Amazon wouldn't be, there wouldn't be a reason to be cutting down the trees in the Amazon to grow soy, because we wouldn't have the market for it, because that wouldn't be a appropriate ingredient for the feed for chickens and pigs. We wouldn't have the central of the part of the US growing as much corn as it is, because we wouldn't have cows, even if they're still in feedlots, eating corn, because it's not good for them and it's not economic. There's more economic ways of doing it. So, yeah, I really think we can completely transform um, our humans' effect on the landscape through nutrient density. Um, and we're just looking at it crop by crop right now, because you have to do the science work before you can seriously engage that market. So we did beef as a stand-in for animals. Now we're going to do wheat as a stand-in for grains, and that covers a large amount of the world's, agricultural land. Certainly apples and pears and peaches and tomatoes and lettuce and everything else, I'm really looking forward to doing. But yeah, I mean, imagine there's no dead zone in the Caribbean. The aquifers in Iowa aren't full of fertilizer and toxins. Kids in third grade aren't dying from cancer. You know, I mean, a lot of things could be very dramatically different in very short order, because it does look like, it's actually more economic to work with nature. Um, we just have to have a market which has a metric that people can use to organize around.
Yeah. Plus, also, I think working with nature, it's inherently just more pleasurable. Like it feels better for the farmer and for everyone involved. I saw Dr. Jonathan Lundgren of Ecdysis one time give a presentation, and he was saying how he's like, I guarantee that I could predict the mental well-being and the happiness of someone based on the microbial diversity that I can find in their soils. He's like, and I think that there's actually not just like a tangential connection there. I believe there's actually a direct connection. And that's also a huge Pandora's box that we don't need to open. But
Well, your gut flora has a major impact on your psyche and your hormones, right? I mean, the what do they call it? The vagus nerve, right? There's
yep,
10 times as much information coming from your gut to your brain as there is from your brain to your gut. Your
yeah,
whole biochemistry is dramatically affected by your microbiome. Yeah.
yep,
No, I
yeah,
think the opportunities for human, health and psychological, emotional, physiological, etc., spiritual health are at our fingertips. At our
yeah.
fingertips. I think,
And I
yeah,
would guess that just like the bell curves that we're seeing, that the majority of us are actually living pretty low in terms of like the total potential that
yeah,
the human potential that
yeah.
we hold.
I totally agree with that.
Yeah,
Bar is low. You know what? There's
yeah.
tons of opportunity for improvement.
That's the good news, right? The bar is
That's the good
low. We got
news. Yeah.
massive areas for opportunity.
If you're happy here, there's a lot of opportunity to get even more happy.
Beautifully said. Beautifully said. Well, I think we can kind of start wrapping with that. I just have a few kind of ending questions for you. One is, what can we do as an audience or agrology, or just sort of, what can we do to help you? And sort of what are some of the blockers that you're currently facing? And how can this growing community help support you and the work that you're doing?
That would usually come from longer conversations. Oftentimes inspired by someone saying, "Hey, I want to do this," or "Hey, this should be done." And we're like, "Yeah, it should be done. Do you want to do it?" We're say, "Great." Um, we're completely grassroots, relationships based network of I think some pretty cool people. Anybody can feel free to reach out. We're pretty responsive. Obviously, I would say, if you wanted to join the conference, I think we're 12 weeks into the 16 weeks. Um, you will be able to attend the last 4 weeks live. You'll get the recordings from the first 12 weeks. That would be a way to economically support us and really get um, a deep dive into a lot of the things that I've only been able to scratch the surface on that we're in the middle of. And probably you'd find some people and some organizations and some companies and allies that you didn't imagine, but like actually could be very powerful for you. We're a membership-based organization. $5 a month is $21 a month, whatever it is you want to throw in. We're grassroots. That's always amazing. Research partnerships. Yeah. Usually they have an internally, they're like, I want to connect for this reason. So I would just trust that voice and feel free to reach out as you see fit. Bionutrient.org is the website, the main website. Bionutrientinstitute.org is our science website. Our YouTube page has hundreds of hours of collected recordings from presenters at the top of their field for more than a decade, freely available on YouTube. We've got social media channels, which I don't know because I don't engage social media, but you can probably find us, like us, friend us, whatever. So yeah, thanks for your time.
Amazing. Um, everyone for attending. We'll send up a follow-up now with the recording and then also probably some links to, at the very least, the website, but also probably the studies that Dan and his team have done. Dan, thank you so much for spending some time with us, and thank you for all your work. We really appreciate you and all of the work that you do to support the regenerative movement, to support a more flourishing agricultural industry, and to support more nutritious food.
Thank you very much. Happy to be on, and looks like you got a great community you're building here. So yeah, let's see how well we can work together in the future. The opportunities are massive.
Absolutely.
Yeah.
Absolutely. Looking forward to weaving more. All right.
Okay.
Thanks, everyone. We'll
Great.
follow up with a nice email. All right.
Thanks a lot.
Bye.